Okay, thank you, Paula, and also a welcome from me. I saw on the registration list, quite some people who had planned or registered to call in from different parts of the world. I myself am based at the Lighthouse European facility in Amsterdam, and I'm responsible for this facility as well as director product line for the company. The topic of today, as Paula mentioned, has to do with container closure integrity. The title is a science-based approach for ensuring container closure integrity of sterile vials. So let's go ahead and get started here. A little bit of context and disclosure before we begin. Lighthouse has a long history in the CCI application area. So more than 20 years ago, we introduced the laser-based headspace technique into the pharmaceutical industry. And very soon, one of the major applications for this technique became container closure integrity testing. So we supply this headspace equipment for CCI testing, and we also have our own analytical laboratories where we support customers on a project basis from the lab, and this includes CCI method development and validation, as well as other types of studies. So that's where we get our knowledge base. The context for this particular webinar is that the regulations and industry best practices around CCI testing are currently evolving. So a few years ago, we were asked to give input to the USP 1207 chapter, which was implemented. We have been closely following the revisions of the ongoing EU Annex 1 revision. We've given CCI workshops at the regulator. And so this leads to one of our objectives, which is to support our clients in meeting any new requirements for CCI. Here's the agenda for today's webinar. We're going to start with an introductory case study. It's an interesting one, the validating CCI of deep cold storage product vials. This has all of a sudden become quite a hot topic during the pandemic. It was already building interest in the gene and cell therapy areas. But what's been interesting is that the lessons learned in this particular application area can actually be applied to putting together a science-based approach for ensuring vial CCI in general. So you see that as a third bullet point in the agenda. We'll also briefly touch on the evolving regulations and what is actually driving some of the changes. And as Paula mentioned, hopefully at the end, we'll have a few minutes for Q&A where I can answer some of the questions that have been coming in as the presentation goes on. So that's the agenda, and let's start with this introductory case study. So CCI during deep cold storage. What do we know? We know that at low temperature, maintaining CCI of a pharmaceutical vial actually depends on several things. And these are several areas where risk to CCI can be introduced. So the vial, stopper, cap design, and fit dimensions. So the combination of the primary packaging components. The process, the capping and crimping process, which results in stopper compression to actually seal the vial. Temperature, obviously, especially if you start going to temperatures that are below the TG of the rubber stopper. So that's the glass transition temperature. And what's been interesting is that studies have shown that a lot, if not most, of these sealing failures that are caused by deep cold storage are actually temporary. We'll get to that in a minute. So the risk to CCI at these cold temperatures means that it's actually very critical to generate robust CCI development and qualification data. And we've actually been working in this area for about a decade. There's a reference there at the bottom of your screen, at the bottom of the slide now, which was one of the first publications that identified and demonstrated the risk in these different areas to CCI for deep cold storage product. And with deep cold storage, we are talking about minus 80 storage. So what actually happens to a stoppered vial at these temperatures? So we put together some animations here to help visualize this. I think pictures can often say a lot more than words. And what I'd encourage you to do is just to watch this animation as I flip through the text and as I recite the text, because some things happen a little bit quickly in this animation. So let's imagine that you start with an air-filled vial, which at 1 atmosphere at room temperature. That's what you see now on your screen. Now, imagine putting this vial into a -80 environment, and for that purposes we'll choose dry ice. Now dry ice creates a CO2 rich environment, and so you see that here, and hopefully you saw that the headspace actually condenses. At lower temperatures the headspace pressure will lower. That's just the ideal gas law, creating an underpressure inside of the vial. So now you have a vial with some underpressure in this deep cold storage environment. Let's say that the temperature reaches -78, which is dry ice temperature. That is under the TG, or the glass transition temperature, of most rubber formulations, which is around -65. When that happens, the stopper loses its elasticity. It becomes brittle. And because the different components are shrinking at different rates, you have glass, you have polymers, you have metal, interface gaps may appear, and it could be that the closure of this vial is lost. If that happens, the underpressure that is inside the vial will quickly suck in the surrounding storage environment, the gas environment, which is in this case CO2. And so the vial fills up with CO2. So cold dense CO2 from the storage environment fills the headspace. So the vial is sitting there in deep cold storage, and it's full of CO2 if it has lost closure. Now, imagine that you now take this vial out of deep cold storage and it starts to warm up to room temperature. Now the stopper will actually regain its elasticity when you get warmer than the glass transition temperature. It expands and it reseals the closure. But now you actually have cold, dense CO2 gas that's trapped inside. And as the temperature continues to rise to room temperature, this gas tries to expand, but because the vial has resealed, it actually creates an overpressure. So you have a vial that is sealed with an overpressure of CO2 gas. So you can measure these headspace conditions as an indicator of leakage, that the vial has actually lost closure during the deep cold storage. So you can directly measure CO2 in the headspace. You can measure depleted oxygen levels because the carbon dioxide has replaced the air headspace. And you can also measure this overpressure that has been built up to verify leakage. And if you monitor this vial over time and it retains its CO2 overpressure, then that verifies that the leak was temporary and only happening during cold storage. But now that the vial is sealed outside of cold storage, what happens here? If you actually try and test this vial with traditional CCI tests, it will not detect these failures because traditional CCI tests really depend on an active leak to be detected. So hopefully this animation gives you a good picture of what actually happens to a vial in deep cold storage, or could happen to a vial in deep cold storage. So because of these risks to CCI, or possible risks at deep cold storage, we started developing in a number of projects a science-based holistic approach, which would help to ensure CCI of deep cold storage vial product. And so this slide is actually a central slide in this presentation. I'm going to list a number of activities which we have defined in this science-based holistic framework. So number one, and these 4 activities here, I would say would be activities during the development phase of the product life cycle. So the first thing you want to do is have an appropriate CCIT method. So, and in this case, headspace is a very appropriate method to detect the leaks that are caused by deep cold storage for the reasons I just mentioned. And you're going to use this CCIT method in your packaging development studies. So then you can, once you have a method, then you can design and execute CCI studies, which will help you choose and qualify the appropriate primary packaging components. So you want to make sure that you choose the appropriate vial stopper combination that actually works at these cold temperatures. And you want to demonstrate that as part of your qualification. You also want to do process development studies. And with process, I mean the capping and crimping process, so the vial sealing process. You want to characterize your vial sealing process and, if possible, optimize it. And you can do this with residual seal force testing. And we'll say a little bit more about that in a minute. Once you have done these types of studies, then you can generate an RSF CCI correlation. So using statistical sample sets and getting these results in your studies, you can build up a correlation to show that your vial sealing process is actually producing good CCI. So this is in the development phase of the product life cycle. These last three activities here are then as you go into the manufacturing phase. So once you have characterized your process, you can actually determine what the acceptable lower and upper limits are for your crimping settings on your production lines and for each different production line. You can then qualify these production lines at these limits for good CCI, and then you know that your process is under control. Finally, if the CCIT method that you've developed for your packaging development studies is not yet validated, you can formally validate that method and then use it in a number of activities such as shipping validation, even finished product batch testing, or end of shelf life testing as appropriate. So those are the 7 activities that we've identified in a science-based holistic approach. What's the analytical toolbox that you're going to want to try and use for this? I've already mentioned headspace analysis for CCIT. This laser-based headspace analysis is described in USP 1207 as a non-destructive deterministic CCIT technique. It measures trace gas ingress that comes into the headspace due to a leak, and it can be used to develop some optimal CCIT methods. And we'll give several examples of that later on in the presentation. So that's for your CCI. For your process studies, residual seal force testing can be used to measure the quality of the vial sealing process. RSF actually measures the strain that a compressed stopper puts on the vial flange neck flange to seal the stopper against the vial. And then that's held, the compression is held with a aluminum seal, with a crimp. And you can actually measure that strain. So there's a corresponding force to that. Sufficient compression is essential for seal integrity. And so this measurement actually is a seal quality test. It's not a leak test. And it's also described in USP 1207. We mentioned polling questions. So I want to stop here and do a short poll. These are the tools that we have suggested in this science-based holistic approach, and I want to get some feedback from the audience, and then we can all share these results to see what everybody is using at the moment. So there are 3 questions. I'm going to launch this poll now, and you will see 3 questions, and I'll walk you through them. So hopefully now you're seeing 3 questions on your screen. The first question is, in which stage of your product life cycle do you test for CCI? And this is multiple choice, so you can answer as many or pick as many options as you want. And again, maybe you don't actually do CCI testing, but you're familiar with what's done in your company. So please, answer these questions as well as you can. So let's walk through the choices here. Do you do CCI testing during package development and validation? Do you do that during process validation? So on the production line, filling, sealing, shipping. Do you do this in manufacturing, 100% in line? Or do you do that on a sampling basis in manufacturing? Are you doing CCI testing on stability, or none of the above? So I'll give you a few seconds here. I can see answers still coming in. All right, thanks for your feedback there. Still a few coming in. All right, I'll give you 5 more seconds here if you haven't filled them in yet. Okay, great. Thanks for that feedback. Let's go to the next questions, and then at the end we can share these results. Scroll down on your polling window there to get to the second question and all the choices there. Which CCI testing technologies are you currently using? Again, multiple choice. You can choose as many as you want. Dye ingress, so the blue dye ingress test, the microbial ingress test, helium leak testing, laser-based headspace gas analysis, high voltage leak detection, pressure decay, or vacuum decay, or none of the above. And I see that people had already started to answer those questions, so it looks like most people have now answered it. And again, if you scroll down to the last question, which is, which test or inspection do you use to qualify and monitor capping and crimping of your vials? So this is on your production lines. Again, multiple choice, visual inspection of the crimp, a manual twisting of the cap test, residual seal force test, no specific tests using standard recommended crimping settings from the manufacturer or from the supplier of the capping and crimping line, and finally, none of the above. Okay, a few people still answering that last question. So I'll wait for a few more seconds here. Okay, 5 seconds, and then we'll end the polling and take a look at these results. All right, so let's end the poll. And then here I can press a button to share these results. So hopefully now you're seeing these poll results. So the first question, in which stage of the product life cycle do you test for CCI? So 62% in packaging development and validation, almost 60% in process validation. During manufacturing itself, you have 22% doing it 100% inline, and then 36% sampling, 60% on stability, 5% says none of the above. Okay, interesting results. So most people do some CCI testing across the product life cycle. Which technologies are you currently using? So the blue dye ingress test with 63% is still the most commonly used test. Microbial ingress test comes in at 31%. Then you have a list of various what's called deterministic methods as they're described in USB 1207. So helium leak testing, laser-based headspace analysis, high voltage leak detection, all around the 30%, pressure decay 10%, and again, vacuum decay in the 30%, or none of the above 7%. Okay, so again, blue dye is still the most used test, but you see that these deterministic methods are quite, well, reasonably evenly distributed there. Okay, well, thank you very much for that polling, and those were interesting results. I'll remove that now, and we'll continue with the presentation. Okay, so about a quarter of you use a headspace analysis, so don't need to go into so much detail here to describe this method. I think most of you probably know it's based on spectroscopy from a laser diode, and that laser diode shines light through the empty headspace. It will detect a very specific target molecule that absorbs that wavelength of light, and you can quantify the headspace gas in the headspace. It's easy to use. It's very robust as an analytical technique, and so it's ideal for developing headspace CCIT methods based on trace gas ingress. And you can see that it's described in 1207 for CCI. It's also been recently described in a new USP chapter 922 for moisture determination, actually water activity determination, but that's a different topic. Residual seal force. So I was actually surprised at the percentage of people using RSF. It seems to be perhaps a little bit less well known, even though it's been around for quite a while. In this picture here on the left hand side at the bottom, you see an uncapped an uncrimped stopper. Once the stopper is actually capped and crimped, it is compressed, and a crimp is put on it to hold that compression. And again, this stopper in the produces a strain on the flange of the vial to seal, and there's a corresponding force in that stopper that you can measure to see how well it has been compressed against the vial neck. And that's called the residual seal force. So now we'll go through some case studies which show application of this toolbox to ensuring CCI. And again, we're going to base this on that slide that I showed earlier of this holistic approach. And the first activity that was described on that slide was to develop an appropriate CCIT method. And what you're seeing here is data from a method development study, actually. And what you're seeing are the CO2 measurements that were made on positive controls and negative controls that were stored on dry ice. And the method was actually simply a CO2 measurement. So after these vials were stored on dry ice, they were taken out of deep cold storage and then measured for CO2. If any CO2 gets inside, that shows that the sample is leaking. And so you see many different types of positive controls here. Starting on the far right, you can see gross defects, a 1 millimeter hole punch through the stopper, a needle through the stopper, and then different sized laser drilled holes. So these are very well-defined holes that are laser drilled into the body of a vial of 10, 5, and 2 microns. And then you see some type defects, which are micro-sized wires that are placed between the stopper and the vial neck. Now, the size of these wires is not the size of the leak, because the rubber stopper, when you seal the vial, will try and seal around that wire. And in general, what we see is that, for example, the smallest wire, 15 microns, that typically acts as a sub-micron leak. So you see results there. And you can also see that both glass and plastic vials were used in some of these controls. And then on the far left, negative controls that were unmodified. And you can see that this CO2 measurement picks up very, very well all these positive controls that are actually open or have a closure defect and are leaking and ingressing this CO2 as was shown in that animation. You can also see that 2 measurements were made on these positive controls, one on samples when they were frozen, so immediately after they were taken out of the storage environment, and also measurements that were taken a few hours later after the samples had thawed. And you can see that even a few hours later, you can pick up these leaking vials with a CO2 measurement. So that shows and demonstrates a method that you can use to identify samples that have leaked in this deep cold storage environment. Second activity in this holistic approach, choose the appropriate packaging components with using the results of designed CCI studies. And here we're looking at a different measurement now. Here we're actually looking at oxygen depletion, and we're looking at data from 10 different vial stopper combination sets that were, again, stored on dry ice, this time for 7 days. And we're measuring how oxygen is depleted. If CO2 is getting inside, the oxygen levels will drop from the expected 20% atmospheric levels. And you see here that at least 4 of these sets have depleted oxygen levels. Some of them have very high failure rates. For example, in set 3, you see 39 out of 50 vials showing depleted oxygen levels. And so this gives very clear and robust data to indicate which vial stopper combinations actually work at these low temperatures. All of these vial stopper combinations worked at room temperature, but not all of them worked in the deep cold storage temperature. And one point to make here is that to get this kind of insight, you really need to set up studies that involve hundreds of samples. So doing a study of 10 or even 20 samples just does not give you the insight that you need to make a good decision. The third activity in that holistic framework, holistic approach, is to do process studies characterizing the sealing process, the quality of the sealing process of the production line. So here you see some RSF data. The blue line is a first series before optimization of the capping and crimping settings. And you see that the RSF values are relatively low. They're a little bit lower than what you would have liked, an average of around mid 30s, 30 newtons, 35 newtons. Which is a little bit low. You'd like to have that a little bit higher, actually. And there was also quite a lot of variability here, and that comes from variability between the different capping and crimping heads. Um, from that data, a decision was made to optimize the capping and crimping settings, and then a second series were capped and crimped, RSF tested, and you see the results plotted there on this graph in gray after optimization. You see that the RSF values are now a little bit higher, which is what was the desired end result, and you also see much less variability. And so this allows a quantitative characterization of the capping and crimping process and optimization. And again, you need robust sample sets to cover all of your capping and crimping heads. You want to build up statistical confidence in the process. And in projects that we have been doing, I say here, hundreds of samples are tested for every capping setting on each production line. Actually, we've done projects where thousands of samples have been studied or measured and analyzed, and you get very good insight to qualify and demonstrate that the capping and crimping process is under control. The fourth activity there was actually to correlate the process with good CCI. So correlate RSF measurements with CCI measurements. And so this is what this data is showing here. What we're plotting here are samples that were prepared over a wide range of residual seal forces on the actual production line. So you see samples that are plotted here from that had residual seal force of 20 newtons. Those are the far left samples, all the way up to high residual seal forces, up almost to 80 newtons. So these were low capping and crimping settings to high capping and crimping settings. These same samples were then put into studies, CCI studies, deep cold storage for at least 48 hours. And then after the deep cold storage were analyzed for CCI or maintenance of CCI, the samples that failed the CCI test are plotted in red. And you can very clearly see that it is, if you do not cap and crimp this vial stopper combination tightly enough, you will not maintain closure during the deep cold storage. Now you can try different vial stopper combinations. So here are the results of vial stopper combination Y. Here you see a different picture over this wide range of capping and crimping settings and residual seal forces. You see that all the samples maintain closure. So this vial stopper combination performs better, and you can qualify and validate. You can design a capping and crimping space, which is much bigger than that for vial stopper combination X, and you can qualify it for good CCI. Again, robust statistical data gives you full insight into the sealing performance of the production line. And when you correlate that to CCI, you can get full insight into the CCI performance of various primary packaging components during the deep cold storage. So here, obviously, you would probably want to choose, at least from a CCI perspective during deep cold storage, you would want to choose vial stopper combination Y. So those are some examples of development studies and the data that you can generate. From these development studies, you can make some very data-driven conclusions. The CCI risk profile that we saw of these primary packaging components, it depends on the specific vial stopper combination. You can also very clearly see that the risk of losing CCI increases as the RSF value decreases. So the capping and crimping process is very important. It's very critical, and you need to make sure that that is under control. Robust science-based development data, and here we're talking about hundreds, if not thousands of samples, enables the choice of appropriate primary packaging components. And finally, addressing activity 5 that was listed in that holistic approach. If you correlate the process data with your CCI data, you can actually define what your capping and crimping settings should be that ensure a reduced risk of CCI failure for product that needs -80 storage. So now we move into the production environment. As you're starting to get your manufacturing lines ready for production of this type of product, you will want to qualify these lines. And so in your qualification studies, you will produce vials using the production process at the determined minimum and maximum capping and crimping settings that you got from your development studies. You will use these vials for a CCI validation study and show and demonstrate that these vials indeed are maintaining and have good CCI. These defined RSF limits that you have from your development studies will also enable a number of different things. You can enable line equivalence qualification. So as you're scaling up your production, or if you need to set up production lines at another facility, you can use these RSF limits as a way to then set up and qualify these additional lines. And you might decide also to use it as a possible IPC to monitor production once you start manufacturing. And finally, activity number 7 in that holistic framework, you can perform CCI testing on GMP product as appropriate. And here's an example of that. Here in this particular case, 4 batches of clinical product had been produced, and the first 3 batches had actually been in storage for a while, and then they changed the capping and crimping process and produced batch 4, which was more recent. And again, here the CCI method that was developed was a simple CO2 measurement for deep cold storage product. And what we're plotting here is actually CO2 measurements on actual GMP product that was taken out of storage for the measurement. And here a method had to be defined, and a test process had to be defined where GMP product vials could not be out of the temperature out of deep cold storage for more than 60 seconds. So you had 60 seconds to make a CCI measurement, to do the CO2 test. And here we're plotting the results of the CCI measurements. And on the left there, you see the results from batch 1, 2, and 3, and you clearly see some samples that have lost CCI and show CO2 ingress. I would also draw your attention to the scale here. That's actually a logarithmic scale, and the high levels there are actually 1,000 millibar of CO2, so that's 1 atmosphere of CO2. Batch 4 showed a few samples that had trace amounts of CO2, actually less than 2 millibar of CO2, which was their reject limit. So here, batch 4 actually did not show any failures for CCI. So again, this was done because those batches were produced, and after the fact, they realized there might be a CCI issue. These robust CCI and packaging development studies and process studies were not done. And so they worked backwards and had to do a 100% inspection on these batches. Okay, so the key takeaway from this case study example: a holistic science-based approach using headspace CCIT plus RSF testing of statistical sample set. What did that do? It enabled data-driven decisions about the primary packaging components, about capping and crimping settings, and this helps to demonstrate and ensure good CCI for this deep cold storage product. Now, this approach here, again, was on this deep cold storage application, but it can actually be applied to general vial product. And that's what we'll spend the second half of this webinar talking about. And we're starting projects now. We're doing projects now where we're supporting manufacturers use this approach, but now not for deep cold storage product, but for simply general vial product that is being produced in sterile production facilities. Now, why would you actually want to consider taking this more robust science-based approach, or this holistic approach? There is an evolution in best practices at the moment in the CCI area, and it's being driven by evolving regulatory expectations. So I just wanted to spend a few slides on that. From project experience that we've done, we see that the regulator wants more or better CCI method validation data. If you're going to register a CCI method, they want better validation data. The implementation of USP 1207 in 2016 is now having an effect on global regulations. So not just in the US, but we see that China and Japan, for example, are drafting new CCI guidance based on USP 1207. A couple other countries here are mentioned as those that closely look at CCI. There's this ongoing revision of EU Annex 1, where CCI was a main topic of discussion. And in the current draft version, you see language such as a scientifically valid sampling plan should be used. Frequency of testing is based on the knowledge and experience of the container and closure systems. Sample size should be based on component specifications and process knowledge. So where do you get this knowledge? The regulator here is implicitly saying that you have to build up this knowledge. Well, it's about generating good data. And so you need to have this data. And to get this data, you need to have good methods, which is, again, why we proposed this analytical toolbox, which enables you to generate the data that will help you meet these evolving expectations. So directly here from the regulator, a slide, and it's even titled Evolution. This is from CEDAR, and here they're talking about container closure test methods. They say that CEDAR has avoided being prescriptive, telling you how to do it, to allow manufacturers to develop tests to demonstrate the container closure integrity specific for their product, process, and container closure system. So here again is the topic of method development and generating the data to show this. Here's another slide, this time from CBER. And on the left-hand side, you see all these different container closure systems for drug product. And then on the right side, you see a whole list of CCIT methods, starting with what the USP 1207 calls probabilistic methods. These are the more traditional tests, and then a list of the deterministic methods. And what the regulator is saying is that you have all these different types of container closure systems. You need to develop and implement an appropriate method which best matches your product container configuration. So again, there's no gold standard CCI test. You need to develop and pick one which is best for your particular product container configuration. You also see language from the regulator and in USP 1207 about validating a method to detect a critical leak. Obviously, that raises the question, what is a critical leak? And this definition is becoming broader now. Historically speaking, a critical leak was one that might lead to loss of sterility. So there's a risk of microbial ingress. Obviously, that's the fundamental limit for critical leak for a sterile product. But you can also, and you have increasingly the types of product that need either critical headspace gases or headspace vacuum. And here, a requirement to maintain headspace gas content or vacuum actually might lead to stricter requirements for CCI. Because in general, a container that should protect against passage of gas molecules will also not allow the passage of microorganisms. So if you actually have a product that needs to maintain specific headspace conditions, then CCI your critical leak might even be more strict than just simply one based on sterility. There is no definition of what a critical leak is. You have to determine that per product. But here again is a case study from the regulator that shows at least an approach or an example of an approach. They mentioned here in the second bullet point that the product submission contained a dye ingress method, and the data that was included was some method validation data that used a positive control, which was a 50 micron capillary tube. This was not acceptable because it was inadequate to simulate a critical leak. There was no argument or data to say why this was a critical leak. So there at the bottom, you see that the firm had to revise the method and include a positive control, which was a laser drilled container having a 10 micron hole, and then some data and justification to support this as a minimum critical leak size. So again, just one example, but in general, it shows that the regulator is becoming more critical for the type of method validation and data that is included in a product submission. And in fact, they are looking at the blue dye ingress test in particular more critically. The regulators, just as in that previous example, want to see more robust validation. And here you also see an example where the regulator is asking for spectrophotometric detection of the color change rather than on relying on a human operator, which will give a subjective test. So really trying to make this as analytical as possible so that you can validate it. And the last bullet point there mentions a positive control should be included at the LOQ to indicate that you're detecting properly here in this test. Okay. So it's clear that the regulators, I think, are evolving their expectations, and they're stated in these examples. So then if you step back and decide for yourself that, yes, we want or need to take a more robust CCI approach, what would be the requirements for a more robust approach? So here are some examples of what you might require in a new approach. You want to be able to generate analytical data. So again, comparing to blue dye, you want to remove any human subjectivity in your test. You want your test to be sensitive enough to detect critical leaks. And this is what you'll need to demonstrate with your method studies. You might say you don't want any impact on the product. You want the measurement to be non-destructive. If you have a non-destructive measurement, this will enable you to generate statistical data because you're not destroying product. And a lot of the non-destructive methods can also give you relatively high throughput. And this will give you the ability to actually quantify the risk in your process. Just like the examples that we showed in the first half of this presentation. And it would be nice if your method is actually straightforward to validate. So when we looked at that list of possible requirements for a more robust CCIT approach, we started to develop a more general headspace approach, which we thought would address or meet those requirements. And the objective here was to develop an approach that was similar to the blue dye test, but actually better. This method must reliably detect critical leaks. And what we developed is what we call a headspace gas ingress test method. And in this test method, we use what's called a CCI test vessel. The samples are placed inside this vessel. You see a picture of one on this slide. The vessel can be pressurized with some pressure of CO2. So you remember in the deep cold storage case, the dry ice environment naturally gave you the CO2 environment. What you can do with the CCI vessel is actually create a CO2 rich environment so that any samples that are inside the test chamber that get exposed to this environment and are leaking will ingress CO2. And you can remove these samples and then use a headspace measurement to look for CO2 inside of a vial, which would indicate that it's been leaking. So here you see a little animation picture of the CO2 being put into the chamber, replacing air, and then ingressing into a vial that has a leak defect. And you can see that this is very similar in concept to the blue dye test, except that you're not putting your samples into this blue dye bath, but you're putting it into a gas bath. And of course, there are a lot of advantages to an analytical headspace measurement, which is rapid and non-destructive, to a blue dye test to see if indeed you're getting ingress into a leaking container. So we wanted to show you also just some example data of how this from a headspace gas ingress test. So you start with a positive control set, in this case glass vials that have a 5-micron hole laser drilled in the body. You can measure these vials after you seal them, stopper and seal them, and verify that there's no CO2 inside. You put these vials into the CCIT vessel and then put them through a sample conditioning cycle. And a typical one that we just used for proof of principle studies is to overpressure the vessel with 2 atmospheres of CO2 for 30 minutes. And then the samples are taken out and analyzed for headspace CO2 content. So here you see the results of this, let's say, simple study. 10 positive controls with 5-micron holes measured for CO2 after this conditioning cycle in the vessel. The scale here, units are in torr, so this is pressure of CO2. And you can see that all of the 5-micron positive controls are detected very easily. There's almost 1 atmosphere of CO2 that have come inside these vials during the sample conditioning cycle. So again, an example that robust CCI data can be produced with this approach and enables science-based test method development. So here you very easily show that you can detect these critical leaks. Again, the comparison between that approach, the headspace gas ingress approach, with the blue dye approach, you're looking at the ingress of a tracer gas, in this case carbon dioxide. You make an analytical measurement, it's non-destructive. In principle, it can be sensitive to all leak sizes. Again, you can play around with the duration of the sample conditioning or the pressure that you put on it. And what's nice about this approach is it can also be described by gas flow physics. And there's a publication there which outlines how we at Lighthouse have validated a computational model where you can actually predict for certain vial size and initial headspace conditions and leak defect size how the gas composition will change in your method. And this is very advantageous because you can use this model instead of having to do a lot of experiments in the lab. You can do some of that work computationally. So more details of that can be found at that reference. Okay, so now let's talk about some very practical things in this approach. We have described an approach that uses a CO2 measurement as the base for a CCI test method. You can validate and qualify this analytical measurement in a framework, in an analytical method framework, and such as the ICH-Q2R1. There are standards, carbon dioxide standards that headspace analyzers use for calibration and also for challenging the performance of the measurement of the analyzer. These CO2 standards are NIST traceable CO2 standards at different values of carbon dioxide. You can take these known standards and execute a qualification study where you will measure these known standards multiple times and get very good data for accuracy, precision, linearity, limit of detection, etc. So here, the requirement to straightforwardly validate the measurement and the CCIT method, this is part of that. Here, again, the messages from the regulator. So if you're talking about test method validation, again, this gives you some insight into what is being expected. The bullet point here that is pointed out by this arrow says a test method is validated if it has been proven through scientifically accepted studies to be capable of detecting a breach in container closure system integrity. Test methods should demonstrate the ability to detect a container breach that is worst case or critical. So again, the message keeps on coming back that you need to generate science-based CCI data using positive controls, which let's say represent a critical leak, to validate your test method. Again, some more practical details of how we're approaching this. So we have what we call a USP 1207 CCIT method validation, where, and this is a very simple diagram which kind of just describes the approach. Again, there can be variations on this. Of course, you want to have these positive controls, which represent both critical leaks and, for example, gross defects, to make sure that you're detecting a range of possible leak defects. You can have these what we call type defects, which are these wire defects that are placed between the rubber stopper and the vial neck. And of course, you want to have negative controls as part of your validation study. And obviously, the goals here would be that you would detect the leaks in all of these positive controls and then report no leaks as part of your method in the negative controls. Here again is some example data, very similar to the simple experiment that was shown before, although here again you see quite a lot of data. I won't go through everything here, but you see different sizes of laser drill defects. You see different sizes of microwire defects, gross defects, negative controls. You also see CO2 measurements that were made at different times after the sample conditioning cycle in this test vessel, 15 minutes, 55 minutes, all the way up to 20 hours. And with this data, you can define a method and say, look, even when you measure samples up to 20 hours later after they've been exposed to the CO2 environment in the test vessel, you can still detect these critical leaks with your CO2 measurement. So again, fairly straightforward to generate very robust method data. Now, how do you translate all of this to actually validating CCI on a general vial filling line? Again, here are some approaches or proposed things you can do in what we call this holistic science-based framework. Number one, again, the vial sealing process, capping and crimping. You can qualify this quantitatively using RSF testing. It might be that on a filling line, you have a lot of different vial stopper combinations. So you can use a bracketing and matrix approach to do a CCI validation of that line without having to do every single vial stopper combination. It's very similar to what's used sometimes in media fill practices, for example. And this approach will allow you to do a study on a subset of vials rather than all of the vials that are filled on that line. Here's just an example. What are some factors that would actually impact a headspace CCIT measurement? So these are the types of glass. So here on the in this diagram, you see tubular glass, you see molded glass, you see plastic. If you're actually filling plastic vials. So these are all different types of materials that might affect the CCIT measurement. So you can break those up, and then you can look at the color of each of these, and then you can look at a min and max size. So what are the min and max sizes for each of these types of vials that are being produced on this line? And so this is an example of a bracketing approach to design a vial CCI validation study on a line that is actually being used for a lot of different primary packaging components. You can also consider other things that might affect your CCIT method. So the different types of container formats, we've been talking about vials, but you might also have other things. We've talked about container material. Your stoppers might also be different. You might have the same type of vial, but you're actually using a different stopper. That could affect the CCI, so you'll want to include that combination. So this CCI matrix bracketing strategy, again, you can use this as part of an overall approach to validate a production line for CCI. Okay, so wrapping up here, what are some key takeaways here? Number one, an evolution in industry best practices for CCI is being driven by actually evolving regulatory expectations. And the expectation is that product and process design decisions should be driven by scientific data with an ultimate goal of having statistical confidence in the manufacturing process, in your finished product. This webinar hopefully has described a holistic approach that can be used, which combines headspace gas ingress CCIT methods with residual seal force testing to ensure that the CCI of sterile vials are meeting this recent regulatory guidance. So with that, I'd like to thank you for sitting through that presentation. I hope it was useful. Again, throughout the product lifecycle, there's many different places where you can generate data as part of this holistic approach. Several are listed here, and these are activities that we also support our customers with. So please don't hesitate to contact us if you have any questions or would need any support with any of these activities. And finally, here's some other ways that you can get in contact with us. And I'd like to go ahead now and start the Q&A session. Paula, I know you've probably been busy answering as many questions as you can during the presentation. Are there any questions left over that we can answer in these last few minutes?
Thank you, Derek. Yes, we have many questions. I have selected some for you to answer. The first one, give me a second. Yes. Can CCI evaluation and process validation be done with empty containers? Is the effect of product on CCI considered important?
Yes, that's a very good question. And actually, it's a question that I think gets different answers. USP 1207 actually also addresses this and says that in cases where either product is very scarce or might affect the measurement, you can do some of this work with empty containers. If you want to see really what the sensitivity of your method is, for example, then I would suggest that you do that with empty containers, which are positive controls having these laser drill defects. So you want to focus on showing that your method can detect a defect of a defined size, and you don't want anything to interfere with that. If you have product in the vial, it is possible that some product could actually clog a defect. And that introduces a very interesting philosophical question, because you have a leak detection method, which you demonstrate will detect leaks. But if a leak gets clogged, there's no longer a leak. So do you actually expect a leak detection method to detect that defect? And so here is where you start. I think it's very useful to use a risk-based approach and ask yourself, well, first of all, in method development, we always do work with empty containers and filled containers, because you want to understand what the risk is that this product can clog a leak. And from that data, you can start building a risk-based argument, which you can use in your final justifications. So okay, there's a lot more to discuss there, but it's a very good question, and I hope that answer gives at least is at least partly answers your question there. And again, it will really depend on the specific product container configuration that you're working with.
Exactly. This question comes from your slide 11, where you were describing the method development all the way at the beginning. The question is related to the storage on CO2 on dry ice. How long would you store it? But also, you showed results with frozen as well as thaw
Mhm.
samples. Can you also discuss a bit the incubation time there? How long did you wait until the
Right.
measurements were done thaw?
Right. So when we develop these methods, we typically have a minimum of 48 hours on dry ice as a storage parameter, let's say, and that roughly matches. I think dry ice is most is used a lot or is often used in transport. And transport is typically in that range, 48 hours to 72 hours. We've also done studies putting samples on dry ice for 24 hours and even shorter than that. The hypothesis is that if a vial loses closure due to that low temperature, that will happen at the moment that those samples reach that temperature. So once you put vials on dry ice, I mean, after a few hours, I mean, they're at the dry ice temperature, and you would assume that the leak starts there. Of course, that uh, there's not a lot of data to show what happens in real time at these temperatures, um, but that's an assumption. Um, in cases where you have product in a minus 80 GMP freezer, of course, you don't have a CO2 environment there. And so I think that one of the slides that showed the batch inspection of GMP product, there a process was designed so that um, samples were taken out of the freezer, transferred onto dry ice, and then kept on dry ice for a minimum amount of time before they were actually measured to see if there any CO2 ingress had occurred. And there, if I remember correctly, our method development studies showed that after 21 hours there, you could easily detect CO2 ingress for that product in those conditions. Um, and again, if you create this under pressure, as was shown in that animation, you will actually get CO2 ingress, within minutes. Uh, so that's roughly the incubation time that you need to think of, hours to a couple days.
Thank you, Derek. I'm getting a lot of questions related to positive controls. So if I try to combine all the questions, I think most of them are heading related to the answer you already gave us about having them blocked by product. So I hope that was covered now. But there are a lot of questions about which type of positive controls can be used, for example, in a method development phase. Especially also there are some questions about microwires
Yeah,
and defects in the sealing area. Can you comment on that?
yeah, yeah. So in the examples that I showed, I think there were roughly 4 types of positive controls. So um, laser drill defects, and those are most useful to determine sensitivity of the method because you have a very well-defined defect and you can show or state that my method, you know, will detect a 5-micron hole or a 2-micron hole. These type defects, which are these micro wires between the stopper and the glass vial, those will mimic some defects in the sealing area, but you have no idea what that leak defect is. In fact, it could be that the stopper seals around those wires and there isn't a defect. And so we have also constructed in our lab a leak rate apparatus where we can make uh, a positive control with these wires and then put it on this leak rate apparatus and actually measure what the leak rate is and translate that to an effective size. And what we see is that these micro wires most often, and again, it depends on the capping and crimping, how tightly you cap and crimp, but you will create positive controls that act like a submicron hole. So, a half micron hole or something like that. Um, so those are 2 types of micron size defects. Then, of course, you have your gross defects, which can be simply a syringe through the stopper, or some kind of microcapillary through the stopper. You can also have a very large leak, like this 1 millimeter punched hole through the stopper, to see if your method will actually detect that. And that's sometimes an interesting one too, because the headspace measurement relies on the gas ingress, and so you want to make sure that the gas doesn't escape before you can actually measure it. So we've included all of those types of positive controls in our studies. And maybe one last thing to mention about this product fill is that we will do studies with the defects that are below the liquid fill and above the liquid fill. And in this vessel approach, because you're using, you can actually overpressure, um, you will actually detect also defects that are below your liquid fill, through the use of overpressure of CO2. When you expose the vials to this overpressure, the CO2 actually comes into the vial and up into the headspace.
Thank you, Derek. I do have one more interesting question. Although you have not addressed this particular topic during the presentation, this is still CCI on deep cold storage, so I think it might be interesting to address it. This is related to cryo storage. So particular the question is if you can detect the changes using using nitrogen monitoring, so really quantify the nitrogen in the headspace. I think we can give now a short idea of how the approach would be in that case for cryo storage.
Yes. Yes, so cryo storage is also becoming a hot topic, obviously, for some of the gene therapies and certainly the cell therapies that need even colder temperatures, so down to -150 or even -180. And here, product is stored in a cryo storage system, which is often based on liquid nitrogen, and there. As the question implies, you create a nitrogen-rich environment. So not a carbon dioxide-rich environment, but a nitrogen-rich environment. Now, you can't measure nitrogen directly because it's an inert gas and does not absorb laser light, but you can look at the oxygen depletion. So very similar to one of the slides that I showed there, as nitrogen comes into a leaking vial that's in cryo storage, the nitrogen will replace the air, and in particular will slowly replace the oxygen molecules in the air. And so you will see a drop in oxygen levels, and you can use that as a leak indicator. So we have done also some studies, cryo studies. We haven't done many as those as with -80. But we just implemented a new cryo storage facility in our lab, which will enable us to do many more of these types of studies with a much higher throughput, let's say. So we will start doing more and more of these. But in general, at cryo storage right now, and I won't go into big detail here, but just to summarize, stoppered glass vials do not seem to work very well at these cryo temperatures. And there are polymer vials that are out on the market, and we have done studies which have shown that these polymer vials are able to hold CCI or maintain CCI at these cryo temperatures. Again, you need to do these robust development studies that were mentioned in this presentation to prove and demonstrate that. But that's a rough summary of what we've seen in the cryo storage application area.
Thank you, Derek. Maybe we have time for one more question. This is related to what is the advantage of headspace analysis in the frame of CCI testing for deep cold storage versus the known technique helium leak test?
Right. So there are some helium leak methods that have been developed to make measurements at the deep cold storage temperature. So that means that you have a sample area that is at -80, so you put your sample there to cool it down. So the throughput of those methods are not high, at the very beginning, but I know that they've improved it now. When these methods were first developed, you could maybe measure around 10 samples per day. You can do more than that now. But the one advantage of headspace is that a headspace measurement takes a few seconds. And well, you saw from the data generated here, you can fairly straightforwardly generate data on hundreds, if not thousands of vials. That's one advantage. A second advantage of headspace is that it's non-destructive. I mean, that gives you the high throughput as well, or it's rapid and it's non-destructive. That means that you can actually use headspace throughout the whole product life cycle, including measurements on actual product if necessary. So one of the slides that I showed was of a batch inspection. That was actual GMP product. And again, in that case, the product could only be outside of the storage environment for less than 60 seconds. And so a method was developed which met that requirement, and you could actually make measurements on batch product. So that's another advantage of headspace when compared to helium leak for this deep cold storage application. It's about it's about generating a large body of data. I think that's the main advantage of headspace.
Thank you, Derek. I believe we have 3 more minutes until the session is over. So I think we can thank all the attendees today, and thank you for all the interesting questions. I will try in the couple of minutes that are left to continue answering some questions. So yeah, that's a thank you from my side. I don't know, Derek, if there's something you would like to add.
No, thanks again for all of you who have taken the time to join us. Again, hope this was useful. If you have more questions or if questions come up later, please don't hesitate to contact us. Our application specialist would be glad to have a conversation with you or to send you some answers over the email. So thanks again, and hope to speak to you soon.